Welcome back friends. This is Dr. Harshavardhan Hilli, Assistant Professor, Department of Genetics and Plant Breeding, PGCA, RPCA, PUSA. So in the previous lecture we had already discussed about the extraction of the DNA and its protocol. And now we are going to discuss about the quantification of the DNA. So how we are going to quantify the DNA, how we are going to assess the quality of the DNA after the DNA extraction. So that all we are going to deal with this particular lecture. So we are having two types of quantification methods. One is based on visual method and another one is based on the nano drop. So by visual method we are going to just prepare the agarose gel and then we are going to run the sample on the agarose gel and we are going to check the quality of the DNA based on the intensity of the bands. So what we are going to do in this method, initially we are going to prepare the agarose gel. Usually 1% gel is preferred in order to check the DNA quality and concentration. So we are going to prepare 1% gel along with the dye, and then we are going to load the DNA samples into the wells. So you can just see here in the picture right corner at the top, the wells will be there, and then you are going to load the samples along with the loading dye. So once you are loading it, you are also you will also be using this DNA ladder just for the comparison in order to check how much amount of base space or the size of the particular DNA sample is there. For that we need to use this DNA ladder just for the comparison. So once we are loading this marker and our DNA sample, so we are going to run this particular gel at 80 to 100 volt in the electrophoresis unit. So once the gel is prepared and once the gel is made run, so then what we are going to do, you are going to take out that particular gel and you are going to observe under the UV transilluminator. Okay, so once you are observing under transilluminator, based on the intensity of that particular band, okay, so we are going to judge its quality. Say for example, the thick and bright DNA is observed, the bands are very much thick and bright. That means you can just say that the DNA concentration is very much high. Suppose if you are getting a very a single band near the well which is clear, that means it is also good quality of the DNA. Suppose if you are getting the faint bands, that means the DNA concentration is very much low. Or else if we are having the smear bands, okay, instead of a clear band, if the smear bands are observed, then we can say that there might be a chances of RNA contamination, or there might be a chances of the DNA that is degraded. So you can just see in the next slides here. So how the actually gel looks like. Okay, say for example, if the extracted DNA is of good quality and good concentration, then we are going to get a good gel picture like this, clearly visible. Okay, so if we are going to make any mistakes, for example, if there is a chances of RNA contamination in the DNA sample that we had extracted, that means the bands will be looking like this, smearing will be appeared because of this RNA concentration. Here we might not have used the RNase treatment, so that means there is a chance of contamination of the RNA. Suppose if the DNA is degraded also during the extraction process, then the gel will be looking like this. In the second picture you can see here. And similarly, if the faint bands are there, very blur bands are there, that means there might be a chances of protein contamination. Similarly, if the multiple bands are observed like this, in the left picture you can see at the bottom, so there might be a chances of partial digestion of the DNA is seen here. Okay. Similarly, if the curved bands are observed in the fifth picture, so it will be mainly due to the voltage fluctuation in that electrophoresis unit. And suppose there are any loading issues while loading the DNA sample into the wells, if you are making any mistake, then some uneven bands will be formed. So this is how there will be a chances of bad gels will be prepared based on the quality of the DNA that you have extracted, based on the protocol that you have extracted. If there are any mistakes, that means we are going to encounter these particular problems. Okay, which are the examples of bad gel. Suppose the DNA extracted is very much good, everything protocol is very fine structured, then you are going to observe the very good gel. So the intensity of the band will also be very much good. Okay, so based on this intensity you can easily judge that how much quantity of a DNA is there, whether it is good or bad. Okay, whether it is very highly concentrated or whether it is not high concentrated. Based on the intensity of the band you can judge. If the bright band is there, very high band, good clear picture, then you can say that amount of DNA is very much high. Then accordingly you can go for dilution of the DNA. So if the bands are very much bright, that means no need to go for a high amount of means you can go for high amount of dilution here. If the bands are not that much bright, that means you can go for very much little amount of dilution to that particular DNA sample. So that's how we are going to do it by the visual method, that is based on the gel. Similarly, second method is there, which is based on the use of this particular instrument called as a nanodrop or a spectrophotometer. So what we are going to do here, we are going to take the DNA sample which is extracted already. So we are going to take it, and then we are going to load in this particular spectrophotometer. Here you can see there are different types of spectrophotometer or nanodrops are available in order to quantify the DNA. So this is the pedestal, we can say. So what we are going to do, we are going to just switch on this nanodrop. We are going to clean the pedestal area. Okay, so initially we are going to load the buffer for just for a blank buffer to check it, and then we are going to wipe it again, and then we are going to load our DNA samples, the extracted DNA samples, and then you are going to click measure. Okay, so the results will be appearing. So you can just see here in this picture the quantification of the DNA. After the quantification, so exact reading of that particular DNA sample, it's been shown here. That means in a nanogram per microliter, so it will be showing. So also it will be showing some graphs and all. So based on the quantity of the DNA that is shown here, okay, the readings and all, so you can clearly come to know its quality and all. So just we will see here, so how this particular nanodrop or the spectrophotometer will be indicating the quantity of the DNA or quality of the DNA. So that means it is mainly based on this the ratio 8:60/280 ratio. That means here the ratio will be calculated by this particular instrument, that is it will be measuring the DNA purity actually. So whether there is a mix of protein or RNA contamination, that can be known by using this ratio. So what does this ratio says actually? This 8:260 says that the absorbance of the DNA at 260 nanometer wavelength will be there. Say so for example, if proteins and other things are there, then absorbance at 280 will be very much high. So if we calculate this ratio, so that means here we are going to get the particular like a concentration of the DNA, whether it is contaminated or whether it is pure. Okay, because since the DNA is actually absorbed more at 260, so we will be preferring high amount of like a 260 ratio, means value, than 280 value. That means absorbance at 260 is usually preferred here. Yeah, when you are going to calculate that one, this ratio, so if the ratio is near to 1.8, then you can say that the DNA is very much good or pure. If so, for example, the value is 1.7 or less, then the chances of protein contamination is there, and if the same value is more than 2.0, then there will be chances of high amount of RNA contamination. Okay. Also, you can easily calculate the DNA concentration manually also by using the formula. So, if you will be having this 8:260 ratio, that means absorbance of DNA at 260 nanometer wavelength. So, if you are having that value, then you can use this particular formula. You can multiply with 50, and also you can go for multiplying with the dilution factor. So, if you are going to dilute and then you are going to check the sample, then you need to use the dilution factor. So, if you are not going for dilution, then there is no need of using this dilution factor. Just you need to multiply it 1, or else just you can neglect it. Okay, so 50 is the actually conversion factor for this double stranded DNA. So if the value is there 8:260, you need to multiply with this 50. So accordingly you are going to get the reading. Say for example, if you are getting the reading more than 200 nanogram per microliter, that means it is a very high DNA concentration is there in your sample. Suppose if it is between 50 to 200, then it's a good DNA concentration. Say for example, if it is very less than 20 nanogram per microliter, then the DNA that you have extracted is very much low. So quantity of the DNA that you have extracted is very much low. That means there is some chances of contamination or some chances of any errors are there while extracting the DNA. So this is what I wanted to actually discuss about the DNA quantification, and which are the two methods, by visual method and by using this nanodrop. So how we are going to interpret the results. So in the next lecture we will be coming across how we are going to prepare the gel and all. So in this lecture I just wanted you to know how you are going to quantify the DNA after the DNA extraction. So hope everything is clear. If still any doubts are there, you are very much free to come to me, or if you are very much free to contact me. I will be giving my number. In the previous video also I had shared my number. So 897105442. So you can easily ping me, or do the WhatsApp message if you have any doubts. Hope the things are very much clear now. So how we can go for DNA quantification and all. So in the next lecture, we will discuss in detail about the preparation of the gel and all. Okay. So thank you very much.